Hey all, so today we're going to be talking about DNA and RNA, and we want to be thinking about how are these things the same, how are they different, how are they used for similar purposes, how are they used differently. And overall, our question is going to be, how does the structure of nucleic acids allow hereditary information to be stored? We're going to be looking at A1.2 and looking at pages 16 through 27. So firstly, let's have a look at the structure of DNA here. And so when we're thinking about the structure of DNA, it is composed of nucleotides, and a nucleotide has three components: a sugar, a phosphate group, and a base. And there are four bases in DNA and RNA both. The bases in DNA are adenine, thymine, guanine, and cytosine, and they complementary base pair with each other. So C's are going to bind to G's and A's are going to bind to T's, and they're going to form hydrogen bonds between those different bases. All the other bonds in the molecule are covalent bonds. It's just between the two bases is hydrogen bonding. And you can see here, adenine and guanine are considered to be purines, while thymine and cytosine are pyrimidines. And so what that means is it's just a name for the structure that they have. Thymine and cytosine as pyrimidines are one circle of carbon and nitrogen. And adenine and guanine, which are purines, have two circles, which are carbon and nitrogen. We've already said that about covalent bonds holding the nucleotides together. And then when we're looking at this, we can see that DNA is two-stranded. So there's this segment here on the left, and then this segment here on the right. And one of these strands is 5' to 3' orientation, while the other is 5' to 3' orientation. And so these are said to be anti-parallel, meaning they're facing an opposite direction. So what does it mean when we say 5' or 3'? When we're looking at this sugar, this here is carbon 1, where the base is attached. And so this is carbon 1, carbon 2, carbon 3. And so the phosphate group is attached onto the third carbon, which is called the 3' carbon. Then carbon number 4 is this fourth point, and then there's a carbon in here in this line, which is the 5th carbon, where the other phosphate group is attached, and this is the 5' carbon. So this is actually pretty important, and it's going to come up over and over again, because everything when we're looking at reading RNA and when we're replicating DNA, or we're making strands of RNA, everything is happening on the 3' end of the molecule. And so this 3' end structurally allows for bonding to happen, and it's quite important overall, and it's going to come back again and again. So when we're looking at the structure of DNA and RNA, DNA is deoxyribonucleic acid, whereas RNA is ribonucleic acid. And DNA, we already saw, has two strands, so they're anti-parallel, and it makes a double helix. They have the bases A, T, G, and C, and it's made up of a deoxyribose versus a ribose. And deoxy means oxygen removed. And so here you can see that there's an OH group here, but no OH group there. And in this molecule, there's two OH groups there on the bottom. And you can see with RNA versus DNA, there's only a single strand, not a double strand. It's made up of that ribose sugar. And then also one of its bases is different. So in DNA, it uses thymine, but thymine is replaced with uracil in RNA. And you can see the structures of these molecules. So uracil, cytosine, and thymine are all single circles, and those are called pyrimidines. And then guanine and adenine are purines. So when DNA is replicating itself, that means it's making a copy of itself, which it needs to do through every process because, well, it needs to do it through every replication of a cell. All of your cells have all of your DNA in them. So it's really important through the process of mitosis that your DNA can replicate itself so it can be in every cell. DNA is, the bases in DNA are uniform, so they occur in all living organisms on our planet. The amino acids that we have that make up our proteins, almost all of them are identical, which suggests that we all came from a common ancestor historically billions of years ago and evolved together on the planet. And What that also means is that we can take genes from humans, cut them out of our DNA, and then put them into bacteria. And those bacteria will read that genetic code the same way, and it will produce the same proteins. And actually, that's how humans make insulin for other humans. DNA is the genetic material for almost all cells, but viruses do use RNA in their cells as their genetic material. However, viruses, because they need a host cell to replicate themselves, are not considered to be living. So back to DNA replication. DNA can be any length, and the bases can be in any order. Because there's 4 bases, if you were looking at a sequence of 2 bases, for example, AA, AG, AC, you would have 4 to the power of n being the number of bases, different possible combinations. So if you're only looking at a sequence of 2 bases, then there's going to be 16 possible combos of the order and the combos of those bases. So there's a lot of variation that's possible within there. And as small changes are made to DNA, that can result in different characteristics in humans, which we'll talk about more in different slides. DNA has segments, which are called genes, and those code for particular information in our body. So characteristics, traits, proteins in our bodies, those are coded for by genes, and they're going to be read by ribosomes out in the cytoplasm. So replication is copying DNA. It's going to happen in mitosis or meiosis. Meiosis is making sex cells, gametes. And so mitosis is used in body cells, which means that, if your skin is replicating itself, it is going to make more skin cells, and all of those will go through the process of mitosis. Meiosis is only happening in ovaries or testes, and it's only being used to produce sperm or egg cells. DNA is super long in eukaryotic organisms. In prokaryotic organisms, it's not. It's coiled up and, it's circular as opposed to linear. And it's not coiled up because of the structure that it has. But in DNA, it needs to be coiled around these special histone proteins so that it can stay packaged properly. RNA is the template to make a protein. And so DNA is too big to come out of the nucleus, which then means it needs to send a messenger from the nucleus to the cytoplasm to make proteins. And that is RNA. It's called messenger RNA. And it's abbreviated as mRNA. So that's going to be copied from the DNA, and that's going to move to the ribosome that's in the cytoplasm. And 3 bases together are going to code for 1 amino acid. Those 3 bases together are going to be called a codon. And that's going to be relevant at the ribosome location. So making DNA, RNA, and proteins. So we just talked about DNA replication. The next stage is transcription. So if we want to be making proteins, we have to make some messenger RNA to get it out of the nucleus. So translation is then going to be taking that messenger RNA and having it read by the ribosome and bringing the protein, the amino acids together, linking them up in condensation reactions to make a protein. Adding new nucleotides or amino acids always happens on the 3' end of the messenger RNA. So messenger RNA is being read 5' to 3'. The way it loads into the amino acid, the ribosome, the messenger RNA loads into the ribosome at the 5' end, and then it starts reading towards the 3' end. And DNA is coiled around these histones here so that it can stay effectively packaged. When DNA replication is happening, it does have to uncoil itself. And then also to make messenger RNA, it needs to uncoil. And if you're an HL, you're going to learn quite a bit more about this coiling process, but there's little tags on here that are called epigenetic tags that can affect this coiling process. And if the DNA is tightly coiled onto the histones, then it's unable to open up, and then it would be unable to make these strands of messenger RNA, and therefore it would be unable to make certain proteins. And that's what's going on when your cells are differentiated. So like my skin cells are not making red blood cells, for example, but my bone marrow is, but it has the same DNA. My liver cells are doing totally different jobs, and their cells look very different than my retinal cells, for example. And so what's happening there is in the liver cells, for example, some genes are turned off that are not turned off in my retina. And exactly what's going on here with the coiling is allowing those genes to be turned off or turned on. Okay, so to summarize, DNA is genetic material that contains the genes that code for specific traits. New nucleotides add on the 3' end. RNA is the template for proteins, which are made at the ribosome. DNA is structurally different than RNA. It has ribose, RNA has ribose versus deoxyribose. RNA is single-stranded, where DNA is double-stranded, and RNA has A, U, G, and C as its bases versus A, T, G, and C as its bases in DNA. All right, that is all for this video. Thank you for watching.